serum matrix metalloproteinase 9 and toll-like receptor 4 levels, are useful biomarkers for diagnosing acute aortic dissection. Acute aortic dissection is a serious heart condition associated with increased mortality and morbidity. Matrix metalloproteinase 9 and toll-like receptor 4 plays an important role in aortic pathophysiology. The aim of the study was to compare the serum matrix metalloproteinase 9 and toll-like receptor 4 levels in acute aortic dissection patients and healthy controls, and evaluate the association between serum matrix metalloproteinase 9 and toll-like receptor 4, and their correlation with C-reactive protein and D-dimer. Individuals with acute aortic dissection were included in the study. Same age and gender individuals were matched and included in the study. Individuals with chronic aortic dissection, coronary heart disease, congenital heart disease, or severe vascular stenosis were not included in the study. Individuals with autoimmune diseases, or severe organ failure, or had malignant tumors were also excluded from the study. The study included 176 patients, in which 88 patients with acute aortic dissection and 88 healthy individuals were included. Serum matrix metalloproteinase 9 and toll-like receptor 4 were measured by enzyme-linked immunosorbent assay. C-reactive protein and D-dimer levels were obtained from the medical records. Multiple logistic regression models assessed the correlation of serum matrix metalloproteinase 9 and toll-like receptor 4 with acute aortic dissection risk. Receiver operator characteristic analysis was performed to assess the performance of serum matrix metalloproteinase 9 and toll-like receptor 4 individually and combined for acute aortic dissection detection. The serum levels of matrix metalloproteinase 9 was significantly higher in patients with acute aortic dissection as compared to the healthy individuals. The serum levels of toll-like receptor 4 was higher in acute aortic dissection patients as compared to the healthy individuals. By multiple logistic regression, it was found that acute aortic dissection risk was independently associated with either serum matrix metalloproteinase 9 or toll-like receptor 4 levels. It was found that serum matrix metalloproteinase 9 was significantly associated with toll-like receptor 4. C-reactive protein was positively correlated with both matrix metalloproteinase 9 and toll-like receptor 4, but D-dimer was not associated with any of them. The study results concluded that both serum matrix metalloproteinase 9 and toll-like receptor 4 levels were significantly increased in acute aortic dissection patients. Matrix metalloproteinase 9 or toll-like receptor 4 levels were closely associated with C-reactive protein. Therefore, matrix metalloproteinase 9 or toll-like receptor 4 levels alone can be a useful biomarker for identifying acute aortic dissection. The combination of both the markers may help rule out acute aortic dissection.